Good evening, one and all present here. On the occasion of 124th birth anniversary of founder president of Shri Shivaji Education Society Amravati, Dr. Panjabrao alias Bhausaheb Deshmukh, Science College, Congress Nagar, Nagpur, in association with Microbiology Society India, organizing a lecture series which is based on the theme Indian Speaking from Abroad. Today's lecture is lecture 17, which is entitled on the topic Human Gut Microbiome, Diversity Studies to Treatment Option. Our eminent guest speaker of today's guest lecture is Dr. Mangesh Suryavanshi Sir, Postdoctoral Fellow, Lerner Research Institute, Cleveland Clinic, Ohio, USA. So on behalf of the organizing committees, Honorable Principal Professor Mahendra Dhore Sir, Science College, Congress Nagar, Nagpur, and Honorable President, Microbiology Society India, Dr. Arvind Deshmukh Sir. On behalf of the organizing committee, I welcome our eminent guest speaker, Dr. Mangesh Suryavanshi Sir, and all the participants of today's guest lecture. So I take this opportunity to introduce our guest speaker of today's guest lecture, Dr. Mangesh Suryavanshi Sir. He has done his undergraduation from Shivaji University of Kolhapur in 2007, his post-graduation from the same university in 2009. After that, he has done his PhD in biotechnology in 2017 from Savitribai Phule Pune University, Pune. Currently, he is postdoctoral fellow at Dr. Aaron Miller Lab, Cardiovascular and Metabolic Sciences Department, Lerner Research Institute, Cleveland Clinic, Cleveland, Ohio, USA from October 2021 to till date. Before this, in his employment status, he was at Sanjeevan Agrotechnics in 2011 as production manager. After that, in 2011 to 2016, as a research fellow at National Center for Science for Cell Science, Savitribai Phule Pune University Campus, Pune. In 2017, as a consultant at D&B Genomics Private Limited, Kolhapur. In 2018, as a lecturer in Yenipoya deemed to be University, Darklat, Mangalore. In 2018, as a research associate at Sikkim Central University, Samdar, Tadang, Gangtok. In 2018, as a science ambassador at Varna Science and Innovation Activity Center, Shri Varna Vibhag Shikshan Mandal's campus, Varna Nagar, Maharashtra. In 2019, as a research associate at KEM Hospital and Research Center, Sardar Mudliar Road, Rastapet, Pune. In 2021, as a principal investigator in SCRB project at Yenopaya deemed to be University, Darlakat at Mangalore. As a postdoctoral fellow at Ohio Lerner Research Institute to till date. Apart from this, he has carried out some research projects which are entitled on hyperoxaluria and gut microbiome, a case control study for hyperoxaluria and gut microbiome as a principal investigator. Apart from this, on the topic diabetes and gut microbiome, Indian cohort study for diabetes for surveillance of gut microbiome. Another project is based on the title Kidney Stone Diseases and Urine Microbiome, a case control study for recurrent kidney stones and urine microbiome. The fourth project is entitled on the precise and accurate examination of bacterial lithogenic urine biomarkers for kidney stone diseases, which is funded by SCRB National Postdoctoral Fellowship at Yenopaya Research Center, Karnataka, India. In his consultancy, he is the science ambassador at Varna Science and Innovation Activity Center, Varna Nagar, Maharashtra, as a consultant at Sanjeevan Agrotechnics at Kolhapur, Maharashtra, as a consultant at Seema Biotech at Kolhapur, Maharashtra, and as a consultant at D&B Genomics at Kolhapur, Maharashtra. He is also the recipient of many awards and accolades as well as grants. He has received Young Investigator Award and won second prize for poster in 2021 at New Delhi, India. Innovative Dedicated Researcher and Excellent Professional Achievement Award in 2020 by Society of Innovative Educationalist and Scientific Research Professional, Chennai, India. Young Scientist Award by Microbiologist Society, India. In 2021, third prize in poster presentation in 2020 at SPPU, Pune, India. He is the recipient of Research Associate Fellowship of the Department of Science and Technology in 2029. Then he is the recipient of Research Associate Fellowship at Department of Biotechnology in 2018. Got first prize at Startup Yatra in 2018. Then he has attended and presented his own work in the 60th Lindau Nobel Laureate Meeting 2018 at Germany. He is the recipient of Young Investigator Award at New Delhi, India in 2016. Recipient of Travel Grant Award at 2015 at Pune, India. Recipient of again Travel Grant Award at in Immunology and Foundation at New Delhi. Then Indian Council for Medical Research, which he has done his poster presentation at Germany, as well as Travel Grant Award at Department of Biotechnology, Government of India. Young Investigator Award, he won second prize for poster in 2015 at New Delhi, qualified as ARS NET examination 2014. Also won first prize in poster presentation in 2018 at Global Innovation Exchange Program, Hyderabad. Recipient of Senior Research Fellowship Award in 2016. So, apart from this, he has many research paper publications to his credit in the reputed national and international journals. He has also presented many research papers and his presentations in many national and international conferences. He has also completed many genomic level as well as metagenomics applied. Apart from this, some basics of extracellular vesicles, then health and disease related studies, as well as some antimicrobial resistance studies. So apart from this, on many courses, he has completed such trainings and courses in his career. He has also some memberships of the professional bodies like Association of Microbiologists India, Microbiologist Society India, National Academy of Biological Sciences, and Biotech Research Society India. So thank you very much, sir, for accepting our invitation to deliver your precious lecture in today's guest lecture series. So on behalf of the organizing committee, I again welcome you and request you to please elaborate your journey from India to abroad to our participants, and then please give your precious lecture in today's lecture series. Please, sir.
Good evening, all. I'm Mangesh Suryavanshi. I did my PhD from NCCS Pune, and I take this opportunity to thank for the organizers and especially Dr. Arvind Deshmukh sir for giving me such opportunity to discuss a hot topic in the current year like human gut microbiome and allied research. So I'm going to take this. Okay, my slides are visible to you?
Yes, sir, it is visible.
Okay, thank you so much. Good morning, friends. It's morning here. Good evening, friends. I'm going to discuss more about the human gut microbiome. That is related to the diversity studies to the treatment options. So human gut microbiome related studies started with the diversity patterns kind of analysis, then to the right now into the treatment options. So actually, I'm a postdoctoral fellow from the Lerner Research Institute, Cleveland Clinic. And right now also I'm working in the gut microbiome plus urine microbiome kind of activities. This presentation, I'm going to talk about three different things like a background of a microbiome, human microbiome, and section second would be a microbiome diversity, especially the gut microbiome for their analysis tools and techniques for the diversity analysis there. third is that what exactly the treatment options right now in a clinical section. And I'm going to discuss about a case study for explaining the microbiome work into treatment option. And fourth one is it's a take-home message for from the human gut microbiome kind of activities. So coming to the human microbiome, so actually this presentation is only, it's more related to the very basic one. So let's say, for example, what are the definitions of the human microbiome and what exactly the diversity and how we are going to deal with the diversity kind of analysis. So coming to the first thing is that what is exactly the microbiome? So we very well known about that till now, we only limited our definitions like this is a collection of all microbial genetic material found in the community of the microbes which are living together. But in the recent years, the concept of microbiome has been changed. So the proper definition for the human microbiome or the microbiome is referred as the ecological community of the commensal, symbiotic, and the pathogenic microorganisms that share our body space and total biota along with its genes, metabolic products in that particular ecological niche. So I'm to say that it's a trans-domain structural diversity, functional diversity, plus their genes, their metabolites, and their interaction kind of activities. So collectively, we called as microbiome. We know the fact that we are not alone. I mean to say, whatever human body have, along with our body or body sites, there are other microorganisms living together with us. Like we believe that as evolution is proceeded for the human, the microbes also co-evolve with the human species. So till now, the work has been started. The first work has been started like a different kind of body sites, let's say, for example, oral cavity, skins, gut, generator tract, kind of ecological niche from the human body itself. So in the previous days, the scientists wanted to look for what exactly coming along with us. So scientists found that more than 100 trillion microbial cells are with us along with our 10 trillion human cells. And up to now, we discovered more than 300 species from our entire human body sites or along with the human various driving sites. And we know the fact that the structural diversity also dominated by some dominant microbial flora, such as firmicutes and other phyla. And till now, we are able to deduce that more than 10 million genes are present from microbial systems. So this is something like a 360 times more than our own genes. Own genes means our human genes. So we are not a single one. So it's related. We are a self communities, not just a human being. So coming to the human gut part, human gut microbiome. So human gut microbiome, it's very good a topic to learn, to formulate a description, to formulate association kind of activities, plus to formulate a kind of a treatment options to us, because we know the fact that whatever we eat, that goes from gut itself. And since a birth now, since a childhood now, we very well know about so whatever the vitamin requirement, since we are heterotroph, right? And whatever the vitamin requirements or homeostasis kind of activities, which are actually driven by the bacteria present inside our gut. So this is a spectacular thing, like what exactly the microbial community are there, how they are establishing, and what they are exactly doing with our health and disease kind of associations. So till now, uh, for the gut microbiota, so we know the fact that since from the intrauterine environment to our last day of our uh, living form, so we are mostly influenced by the bacteria itself. So I'm here to trying to take up a look that what exactly gut microbiota influences, or what are the environmental factors that influences for the gut microbiota colonization since from the birth to the last form of our life. So a researcher have been found that intra uh, uh, intrauterine environment is not sterile, so that may be a striking feature, or that be a colonization factor for the gut microbiota started before our birth. And next would be a birth mode, maybe a C-section, maybe from the regular uh, birth. Then comes to the infant microbiota, wherever we feed on feeding practice like milk feeding or formula feeding kind of things with us. Next one is that what exactly the weaning phase, which are actually contributed to our structural diversity of the gut microbiota. Then to the most important thing is that host diet, physiology, status of the immune system, plus lifestyle, genotypes, and certain kind of environmental factors who actually have a major impact on our structural diversity uh, into the human gut. So coming to the main part, so this is like a milestones, the different milestones in reference to our human gut microbiota kind of analysis. So so I have mentioned here some milestone till 2017 here, because after 2017, this uh, this microbiome science is evolved far more than our thoughts. So the first publications, the milestone publication is like from the key Nature in the Nature itself. So they firstly mention about the human gut reference catalog, then to the enterotype analysis kind of things, then to the association with the type 2 diabetes. And in 2016 only, Olaf Peterson also published a paper. This is related to the microbiome and the insulin resistant patterns. And after 2017, wherever there is a consensus agreement, like a standardization of microbiome studies has been happening. So collectively, after 2017 or up to 2017 or 2018, so this is a survey, whatever I'm mentioning here. So 29 patent was applied or filed based on the gut microbiota in the 2017, 2018 kind of things. So after 2017, as of now in 2022, there is one fact that whatever the let's say, for example, the funding sources are for the microbiome kind of activity studies is far more than we are exactly thoughts for now. So, and we believe that right now for the microbiome studies, this is the field who actually has the highest level of funding right now. So coming to the exact part, what is exactly microbiome do with us? So whatever we eat daily for our energy purposes, it comes to the gut itself, then goes to the colon. It gives it metabolized by the host as well as metabolized by the gut bacteria. They forms different kind of metabolites, and whatever it is, the form metabolites, soluble metabolites, insoluble metabolites, various kind of proteins, molecules, or digested part that goes into the body, and it gives an we can refer it as a healthy gut. What exactly normally happening inside the gut, but wherever there is kind of disturbance happening, let's say for example, any kind of disease mechanism like chronic kidney diseases or Crohn's diseases, IBDs. Let's say, for example, if you are suffering from any kind of anti-nutritional factors kind of ingestions, and everything is there. So wherever there is a change, that change happen inside the gut itself. So it's very limited, very short limited time point. This is acutely disturbed gut. And when it is consistently along with the time point, we call them as a chronically disturbed gut. So this is the fact that we wanted to check what exactly the dynamics happening with every time point with the that time slots or that kind of distribution patterns. So in a recent years, we able to formulate the fact that one kind of a dysbiotic environment. So in the ecology itself, we in the gut microbiome or microbial science studies, we adapted much more definitions from the ecological backgrounds. So the ecology related, like whether here I mentioned about the gut dysbiosis. Dysbiosis is kind of it's kind of a disturbed kind of activities. So wherever there is a disturbance inside the gut itself, so there are some relations happening there. So whatever the bad bacteria are being dominating over the good bacteria inside the gut. So this is like something a puzzle is solving with the help of different kind of diseases with the human being. So in a recent years, as a researcher now, so we are into the process of developing the correlation kind of activities in the disease stage to the human gut microbial diversity. So we, being a researcher, we wanted to formulate a correlation kind of activities, whether this is happening through the disease itself or from the disturbed gut. That is actually the origin for the comorbid state of the human body. So till now, in a literature itself, we found very good correlation between the periodontial, non-alkali fatty liver diseases, IBD, immunological disorders, arthritis, obesity, insulin resistance, cardiovascular diseases, even into the Alzheimer's diseases. So we are able to link our gut bacteria to a different kind of diseases here. So in a previous day, we are able to formulate a correlation like dysbiosis condition. So in the dysbiotic condition, disease specific flora is going up, and whatever the healthy related flora is going down. So this is not something like a mere correlation kind of activities, but in intestinal and extra-intestinal kind of two diseases, we have a clear-cut communication or clear-cut definitions or clear-cut associations between different kind of bacteria. So this is something like not related. We wanted to check what exactly the structural diversity is there, but whatever the functional flora, let's say for example, functional phylotypes are there, can be involved into the such kind of a disease perspective or not. So, um, we know the fact that wherever we wanted to think for the any kind of disease, we need to analyze first what exactly the structural and the functional diversity changes there. So I'm going to discuss about how we are defining the microbial diversity with the help of different tools and technique right now available with us. So we know the fact that so wherever there is a healthy flora inside our gut, and wherever there is an onset of that disease, so it may be due to the it's a cause or the consequences of that microbial structure. But right now we are able to find out the disease cause in terms of diagnostic. So we can manipulate the disease condition through the diet itself. And we wanted to look up as a microbiota, can we see whatever the observations we are with us, so can we treat the disease with the help of microbiota? So in a current year, whatever the research is happening, so the research is only is right now focusing on the whatever the microbiota has been changed. So at a certain point, can we modulate the microbiota in the disease perspective to the original one? Is like can we reversal the microbial flora to the healthy one? So most of the problems will solve. This is the best assumption. This is the best option to us to look for the microbiome kind of studies. So in a recent year, the very popular tool for studying the microbiome analysis with the help of genomics. So, the most popular way, like extraction of the genomic DNA from the environmental sample. And we have three most choices, like amplicon sequencing, functional metagenome, and complete metagenome. So amplicon sequencing, when we exactly correlated with our ecological niche, with structural diversity as well as the structural diversity mostly. So we choose for the 16S RNA gene. If you really wants to look for the functional kind of annotations of functional gene involvement or functional flora involvement, we can able to do that with the functional gene amplification. But this is the most popular or most appropriate thing is that the complete metagenome kind of activities. And for the fourth step is that the computational analysis, statistical analysis, data storage, and the sharing kind of things. So I want to particularly mention here that whatever the analysis is related to the microbiome work. So this has been trending towards the computational kind of activities. So we believe that and we know the fact that whatever the data is coming up from the any ecological niche is into the GBs and in terms of TBs. So we need that power, we need that logic, we need that that level of that level of analysis system. So this is more trending towards the computational kind of activities right now. So in the more about in the diversity patterns like alpha, beta, machine learning approaches, co-occurrence studies, and the metabolic networking kind of activities are the prominent one for microbial diversity analysis kind of activities. So I said that the DNA is the most a popular way to analyze the microbiome into the any kind of ecology. But we know the fact that there are the central dogma. We know the system like a DNA to RNA, RNA to proteins, proteins to the metabolite synthesis kind of activities happening in any kind of living system. So for the microbiome also. So we can check we can with the help of any different kind of material, let's say for example DNA, RNA, proteins, we can take that material, and based on that material we can do the tools like genomes, transcriptome, proteome, and the metabolome kind of activities. As I mentioned in the genomic kind of activity, the most popular one. So the technologies or the analysis refer to every different kind of products we are using. So that is referred to the metabolome is used for the analysis, the metabolomics. If the RNA used for the analysis, we can say as the transcriptomics. We know the fact that genomics use of DNA material for the genomic analysis. When we look for the protein-related activities of protein as our target point, so this is called as a proteomics applications. So in fact, that based on these techniques, we could able to find out the questions like who are there exactly, how they are doing, and what exactly the associations between the any ecology and the microbiota present there. So in a recent years, so we are adopting the 4 different, it's like a omic approach, like a metagenomics, metatranscriptomics, metaproteomics, and metabolomics. Meta stands for the collectively all the microbial or all the system present there. So this is not regarding like we are targeting only the bacteria. It's more about the transdomain kind of activities. So if you look for 16S RNA gene sequencing, you are targeting only the eubacteria, but in the metagenomic kind of activities, you actually going with the transdomain. Transdomains like the bacteria, fungi, archaea, viruses, all those things. So like metagenomic also, from the metatranscriptomic, metaproteomics, and the metabolomic activities. So you can actually integrate those techniques into the single forms. So this is the factor right now in our research, like a microbiome science. So we have to target like not for looking for the only the diversity pattern. We are also in the situation like to analyze the questions arising from that microbiome science. So this is like adapting to the integration of the multiomics techniques into our gut microbiota studies. So that was in the first approach, right now very well adopted nowadays in 2022. And we have another options like as I told you, we are more towards the computational kind of activities. So use of computers to train the computer how and what are the microbiota actually involved into the disease parameter and the healthy parameters concept. So in a recent year, so our machine learning approach or use of artificial intelligence, we can train our computers to with respect to the disease perspectives, and we can involve that kind of approaches for the new diagnostic tools. So in a current situations, so whatever the microbiome catalog with us, so in a bioinformatic way, we can divide into the health associated and another is the disease associated flora. So whether we can segregate those flora and use of those flora through the bioinformatic analysis. So let's say, for example, if you identify the healthy associated flora, when treated, use of that flora in the intervention of the disease experimental animals. So wherever there is a change happens or the resilience happens to the healthy flora, this is like an experimental model we generally are adopted nowadays. So So we call them as a neo course postulates for that. So we know the fact that in the microbiology itself, the course postulates are the crucial factor for disease specific flora identification. So so we are adopting the new coach postulates for such kind of activities. So coming to the third part is that whether gut microbiomes, gut microbiota or gut phylotypes, or the entire gut microbiome has been an option for the treatment or not. So I'm going to tell you the fact that actually the bacteria who are actually present inside the gut who have the ability to exactly treat the diseases as like other drugs we do. So in a recent years, so there are two factors like FMT. FMT stands for the fecal microbiota transplant. This is the whole community extracted from the gut itself. That whole community can be useful for the treatment options, or the microbes. It may be single or into the consortia that gives an a treatment option. So I am going to tell you what exactly the logic behind that and one case study. So uh, the last week only, uh, uh, last uh, last week only, November 30, we have a good news for us because we are dealing with the gut microbiota. And in the US itself, the FDA first time approves the fecal microbiota products or FMT as a treatment option for the prevention of recurrence of Clostridia difficile infection in the adults itself. So this is really encouraging to us, and this is a very good science or very good uh, approach for us to look for the microbiome science for the treatment options. So this is like an immense pleasure to work on the microbiology. Being the microbiologist, it is our own. This is our own factor, like whether whatever we are doing for the research itself, can we really do the real science for the treatment options or not? So, um, Ribaita, a kind of FMT FMT product, has been approved now with from the FDA itself. So we know the fact that it's around 90% of the recurrence rate has been controlled through the Ribaita. So we know the fact that Clostridia difficile infection is it's nearly non-treatable. So so prevention of the recurrence is our the high priority nowadays, because Clostridials are into the fact like they are the more resistance to the antibiotic treatment. So this table shows us that the different kind of countries and their associated approvals. So in the recent year only, USA approved for the FMT or gut microbiota derived treatments. Derived treatments. Right now it's adopting here, but in a certain kind of countries like Belgium, Denmark, they are far more ahead. Like they are able to make an let's say for example able to treat the disease with the not only by the gut bacteria, they are able to treat like with the the proteins, their metabolite products, and their extracted materials can be available for the treatment option. So they are more advancing the microbial science nowadays. So coming to the coming to the FMT part, so this is like a one kind of illustration here. So Serious Therapeutics, it's a company who actually into the microbiome science and FMT kind of activities for the different treatment options. So I'm going to tell you they have 5 different kind of formulations here: SER 907, 287, 401, 301, and 155. So those kind of products exclusively derived or exclusively utilize the gut bacteria for a different kind of diseases. Diseases like Clostridium difficile infection, ulcerative colitis, even into the cancer treatment, plus the other kind of infections like bacteremia and other kind of complications. So as a case study, I'm going to discuss like what exactly that product is, because this presentation or this kind of case study is into the public domain. So I have choose for this presentation. So see the logic here. So what they mention about the strong scientific rationale. So they firstly know about what is exactly the rationale behind that. Yeah, in the publication or in the public domain, they've found that spore forming firmicutes bacteria who are actually prevent the Clostridia difficile germination and the growth. Okay, so so they have prepared one formula. It's a consortium formula who actually involves the spores that are resistant to gastric acid and which are facilitated to the oral delivery to the GI tract. So such kind of microbial consortia can be used as a treatment of the Clostridia difficile, and which is approved now for the drug designation kind of things. So here is the logic. We know the fact that due to the antimicrobial resistance pattern. So let's say, for example, wherever there is a Clostridium infection for the humans, so they are undergoing the treatment in terms of the antibiotics treatment. So wherever there is antibiotic treatment, whatever is the Clostridia difficile vegetative form of bacteria inside the gut, they are treated or they are diminishing with the help of antibiotics. But at a certain repeated kind of things, the recurrence rate is high because Clostridia difficile, those are the bacteria have a spore forming bacteria, and we know the fact that spores are very much resistant to the antibacterial treatment. So here is the logic that whether we can make an consortia who can actually activate that spores to the vegetative forms, and after that vegetation form, we can treat that bacteria with the help of antibiotic treatment. So recurrence rate can be going down. So this is like a logic, and here they mention about a consortia bacteria, a consortia who are the firmicute kind of flora, and a spore forming flora, the healthy flora who have the ability to induce the Clostridia spores inside the gut into the vegetation form. And for the vegetation, whenever a Clostridia difficile into the vegetation form, that can be treated with the help of antibiotic treatment. So this is the simple mechanism. I mean to say it's an indirect mechanism where those kind of flora who activate the spores to the vegetation form, and that vegetation form can be treatable. So this is the indirect mechanism they are highlighting. So let's say, for example, they mention about the oral bacterial consortia. It's 109, SERA 109. So it's a formula consisting of the different kind of firmicutes, spore forming firmicutes into a capsular format. It goes to the gut. Actually, those are the flora, actually the healthy flora, and who are diminishing into the disease perspective, like a Clostridium infection. So they have chosen that flora who are actually involved in the disease perspective. Whenever there is an ingestion and coming to the gut itself, so they start germinating. And whenever there is a germination happening, along with their own germination, germination of Clostridial spores has been happening. So they also mention about the pharmacodynamics activity here. So that flora also have the major contribution to restructure the exact microbial composition inside the gut itself. So wherever there is a change of a microbiota and the function has been changed. So let's say, for example, the more of the bile acid synthesis, short chain fatty acid synthesis, tryptophan metabolism, and other kind of activities has been happening after such kind of flora. So this is like a fabulous mechanism. So this is like a fabulous mechanism. Right now, whatever our lab or Cleveland Clinic is working on the direct mechanisms, like right now I'm working on the hyperoxalular conditions, hyperoxalular kind of patients. So our direct mechanism is that we are now able to find out exactly whatever the hyperoxaluria condition is actually driven by the oxalic acid inside the gut or into the circulation system. So we wanted to develop right now who are the bacteria actually involved into oxalate degradation kind of activities. So this is more like our agenda, like to treatment of that hyperoxaluria or hyperoxalular condition can be reduced to a normal level with the help of actively metabolizing bacteria. So coming to the last part is that key human gut microbiome for the how we can take a good message from it. So so I mean to say that there are two different phases of human microbiome kind of things. Let's say, for example, the bacteria have the positive effects and the bacteria have the negative effects also. Let's say, for example, we know the fact that the bacteremia is into the circulation is being driven by the invading bacteria itself or pathogenic bacteria itself. But in a contrary, the more about the positive side. Let's say, for example, immunomaturation and functional activity actually driven by the gut bacteria. Behavior and brain development has been happening through the gut bacterial metabolites. Protection and the clearance from the pathogen invasion inside the gut, which is the primary kind of activities, along with the capacities to digestion and the nutrition kind of activities through the gut bacteria. So those are the very good topics are open up in a current year. So this is like a ocean to us. So we can choose anything. So this is a very good example, like in Indian communities or from the India itself. So one literature suggests us that there are other scope of the microbiome studies, like we do with the population studies, like pharmacokinetics or drug availabilities kind of things. Microbiome in the health and disease, like we generally do with the diagnostic biomarker kind of activities. Most important thing is that maternal and the child microbiome. So can we exactly look for the maternal microbiome to the fetus development, for the development of healthy, sustainable, whenever the child into the fetus format. And more important thing is that their genes, genomes, and metabolic products for the microbiome can be used to treat a disease, and those kind of scope have been has been calculated or defined by this from the NCCS group. So, so at last, we have a take-home message like current research suggests us that every organ has its specific flora, even sperms, unforeseen organs, even the cancerous organs. So this is the current research. We can thought about any kind of organs. So that organ have their own specific flora right now. So another important thing we should know, like whatever your weight, you have your weight like you have a 48 kg, you have a 82 kg. So 2 kgs of that weight has been contributed by the microbiome itself. So your weight is not your self weight. You can count your weight your weight contributed by 2 kgs by the microbiome itself. Third one is that all treatment options start with the gut action. Even whatever you do take the drug, let's say for example paracetamol, whatever you take a drug, that efficiency has been fully or pharmacokinetics of that drug or efficacy of that drug is a more dependent on the gut bacteria. So this is the real science exactly. So in 2022, we have a clear-cut idea about whatever the circulatory metabolites into your blood or into the plasma. So 60% of that that metabolites from the plasma itself has been coming from the microbiome origin. So this is very encouraging to us. So the latest option, as I mentioned about that, if you are really looking for accessing for the microbiome, you should choose for the multi-omics approach. And for the causality purposes, we adopted like a neo-cause postulate for such kind of association studies. Sorry. And the functional aspect of microbiome, which are currently working in all over the world. So in our department, in the cardiovascular department also, Professor Dr. Hazen is also working on how we can modulate the cardiovascular events with the help of gut microbiota or their derived products. So Dr. Stanley Hazens is working on such kind of very fabulous activities like the causality rules of the TMA to the cardiovascular events. So this is a fantastic or the ocean of the knowledge into the microbiome kind of activities. So this is a good chance to look for the gut environment or the microbiome kind of activities for our research. So that's it. Yeah. So thank you.
Okay. Thank you very much, sir, for your very informative lecture on the topic human gut microbiome, diversity studies to treatment option. So it is really very elaborative and many more things we are getting aware about the role of microbiome in treatment option. So I think participants have some queries. They can please ask to our eminent speaker about the topic if they have any.
Hello, sorry, can I have one question if you don't mind?
Yes. Yes.
Oh, thank you.
Please.
Thanks, Dr. Mangesh. It's fascinating presentation and it's very informative. Thank you. We have been working on probiotic bacteria, and also we got a few publications. Currently, we are working. I'm also leading nutraceuticals and toxicology. for undergrad program. Yeah, it's this topic. I visited New Zealand, and we have seen in vitro model for gut in one of the labs. So we are also interested to recreate, to see, and also in Indian population we have in a food system in our so we are using probiotic, for example, dahi. And mainly in South Indian communities, they use almost on a daily basis, including me. So without yogurt, I can't sleep in the night. It helps a lot. So, and also how that will interact with gut microbiome, and that we are interested to do some more research. And I'll be in touch with you to establish some collaboration. Thank you. It's fascinating update for me. So thank you. Thank you.
Yes, sir. If any participant have any query, please ask. You can please ask.
Yeah, myself. Hello.
Yes, please.
Thanks, Dr. Mangesh, for this nice lecture. I have one query regarding, like you people are working on the diversity of the microbiota. So my question is that there are many complex diseases like UC, IBD. You mentioned that cystic fibrosis. These diseases are caused by defect into the microbiota. There are dysbiosis of the microorganism into the gut condition. So I'm asking, is there any metabolite or microbial species we can identify that can act as like a indicator for that disease? Are you working in that area also? Your suggestion.
Yes, we have very much identified the different phylotypes who are actually culprit for such kind of activities inside the gut itself. So if you are looking for the ulcerative colitis or IBD kind of activities there, so there are clear-cut relation between the Faecalibacterium species and other phylotypes are there. And I'm sure you will find out you will really find out the treatment options for that. Yes, that research is ongoing right now.
Okay, and that FMT, what is the success rate of this FMT, sir? Because like into the gut, the population of the bacteria is like 10 to power 14 CFU per ml.
Right.
And sir, if we if we introduce the bacterial species into the gut, already the species, the concentration is so much high. How they make a space for the new entrants? What do you think, sir?
Okay. So I'm not the right person to answer these questions, but I can give you any suggestions. Can I give you a suggestion?
Yes, sir. Yeah, please.
Okay. So my sir's friend, my sir is Dr. Yogesh Socha from the NCCS Pune. So his friend from the AIMS New Delhi, Vineet Ahuja, who was actually involved into the treatment options for the ulcerative colitis and everything. So can you please approach him for the particular questions like he? So yeah, so FMT for the ulcerative colitis and IBD kind of diseases, actually he is right now working on that. Actual mechanism of that.
We have preclinical trial for ulcerative colitis using Bacillus licheniformis. And actually, one of the lab in US, they in fact asked me that culture to study clinical trial in Bangalore. We are actually working on it. Please go ahead.
Nice, sir.
Okay. Thank you. Thank you, Dr. Mangesh. Thank you.
I think.
Sorry, can I add one more point? Sorry. Dr. Krishnan's question related to secondary metabolites, and also Dr. Krishnan mentioned about the cystic fibrosis. We worked on Pseudomonas aeruginosa for cystic fibrosis and the pyocyanin, one of the secondary metabolites. You can look at it. And also we looked rhamnolipid produced by Pseudomonas aeruginosa But it's not on a gut microbiome side, but mainly from sputum and other samples, we can characterize and identify these compounds. So if you need any information, I'd be happy to share some papers from our research group. Dr. Krishnan, I can share my email address after this talk, so you can contact me. Thank you.
Thank you, sir. Thank you.
I think Dr. Sharath Kamble sir wanted to ask some query.
Yeah,
Dr. Sharath
thank you, Dr. Pranita. Thank you, Dr. Mangesh, for a nice lecture and very nice, like brief introduction. Whatever you done the work with microbiome, human microbiome. So my question is, have you come across anywhere in your studies that have you studied actually? Is it any relation with the ulceritis or like colon ulceritis or colon cancer, and is it related with some microbial organisms like human microbiota? Is it any changes in that particular patient, etc.? Have you studied like that? Or and other question is like related with that, if any diseased person is there, how you identify, because each person has his own microbiome in his gut, yeah. So how you connect with the diseased person with the healthy one? I mean, like how you differentiate with the two individuals. And the other question, as my previous question is, have you studied or have you focused on any relationship with this disease condition patient with the cancerous condition of colon?
Okay. Thank you for the question, Dr. Sharath. So this is like a very interesting topic you are asking me. So in our Cleveland clinic, the RAISE lab actually working entirely on the colon-related morbidities, including the colon cancer, and their associations between the gut microbiota kind of activities. So I'm hearing their updates during our presentation sessions. Like they actually formulated such kind of activities, like what exactly disease perspective kind of bacteria involved into the colon cancer itself, and what exactly strategy they are going to adapt. So such kind of activities, like they have mentioned about key 3 different kind of activities can be used as a treatment option. One is that key inhibition of that pathogenic flora at a first place whenever there is a recurrent kind of activity, recurrent kind of that cancerous cells is there. Another important factor is that key how we can make an use of a different kind of antimicrobials, not antibiotics, to exactly diminishing that kind of involvement of that specific bacteria into the cancerous cells. So they have a plenty of ideas, right? Yes. And your second question is that key how exactly they are doing. So as I'm telling you that key the current research going with the multi-omic approaches, but for such kind of studies like cancer studies, or let's say, for example, it's like a co-evolution kind of activities with the disease perspective, like a specific organ studies. So single cell multi-omics approaches they are adopting. I mean to say, whatever we are doing right now, I am doing it's a multicellular level, or it's a metagenomic level, or it's a metagenomic or metatranscripting level. But right now they are adopting like a single cellular level. So in a single cellular level, how the metabolites, even the whole cell or the metabolites or their other synthesis product actually involved into single cellular level. So this is really fascinating, right?
Thank you. Thank you.
Okay.
Thank you, sir.
One or two comments actually I made. I believe some time is there to interact or discuss. Dr. Pranita, is it okay? I actually put in the
Yes,
chat
sir.
box, so maybe the last.
Actually,
to read.
sir, yes, sir, I am unable to get visible that messages in the chat box because of some technical issues. So, sir, please, you can please ask the questions directly.
Yeah. Actually, just first of all, I would like to congratulate Dr. Mangesh for his such a nice work and long-standing expertise in this area. I know him the time he was working with Dr. Yogesh Soche, and then subsequently at Enopoya University in Mangalore. And of course, now what the work he's doing in Cleveland. I had quite good interaction with him a year back when we were preparing a proposal for the gut microbiota and kidney diseases. Just I was actually wondering, my Mangesh, when we give this high doses of microbial cultures through FMT, what is the response on the human body? Whether everyone accept that everyone's body, or then you find some sort of any reaction or some any kind of consequences. So that is what actually I just wanted to know one thing. Can you hear my question?
Yes, sir.
Yes. What is your
Yes.
opinion?
Yes. So in a current situation, the term is like a personalized medicine.
Okay.
Right. So your questions is more related to that. So whatever is uniqueness to the genomics, let's say, for example, we believe that the human has its unique genetic makeup, or their metabolic response, or their unique metabolism power, right?
Yes.
So likewise only in a current era, with the help of let's say, for example, with the help of artificial intelligence or machine learning approaches, we can easily formulate a personalized medicine kind of activities there. Let's say, for example, whatever your question is, that whether we can wherever there is a differences in the population and
exactly.
Yes.
When we treat a large number of population, to what extent it is acceptable, and what are the cases where you can say it rejects, or no, it is not working properly, efficiently. That is what actually I just wanted to know. No.
Absolutely right.
Was, when we look at this firmicutes against Clostridium, do you really look that they should have the inhibitory activity, or in the anaerobic condition they work? What is your opinion?
So they are taking like aerobic cultures.
Yeah. So
and most of them, yes.
And then Clostridium, we see that it is anaerobic organisms.
Yeah.
Like yes. FMT, okay, for IBD or IBS, it works. Any other cases are there where it has been used?
Yes, sir, of course. Right.
I think the one of the example which you are giving, it is there.
Right, sir, right.
Like
yes.
the microbiota transplantation. I believe for IBD cases, it is successful, right?
Right.
For any other cases treatment?
Yeah. So the thing is that whatever you are talking about, the disease perspective or kind of things, but whatever your questions, this is more related to the anti-nutritional kind of activities or homeostasis kind of activities. So we have another area where such kind of abilities, such kind of activities can be more suitable there. Let's say, for example, what I'm doing right now, it's like the anti-nutritional factor is the oxalic acid present inside the gut. Yes. And we are looking at the harnessing the power of microbes who have ability to degrade the oxalic acid into a reductive level. It's simply we call it the homeostasis of that oxalic acid into the circulation. So I think is that personalized medicine or personalized kind of treatment can be there. Can be adapting to the such kind of nutritional aspects there. So I can tell you a case study like in the recent years. So it's like the companies like Digby Health from the USA, they are able to find out the gut microbiota, their differences to the gut microbiota that is exaggerated to their health status. In status, let's say, for example, cardiovascular phenotypes or insulin phenotypes kind of things, or diabetes status there. So with the help of that microbial signatures, they are going to prescribe the personalized medicine, like what kind of nutrition you are required, and what kind of diversity pattern change you required for that. So those are the answers are coming up in the in the commercial values also. So this is a fabulous thing. So I want to add one more thing is that for the personalized medicine kind of activities. So right now we are adopting a highest technology here with the IBM. IBM only. So IBM is the producer of the quantum computer, right? So they have installed the quantum computer in our system. So right now we are planning to do such kind of the massive work on the quantum computing.
Yeah, just one more question. When we treat the disease by any medicine, we have suppose a week or 10-15 days course to take the medicine, right? Especially with the probiotics, we have always a preclinical trial of 1 month to 2 months. If we see through the gut microbiota route, how many days you think we can actually go on consuming or looking for changes? How many days one should you have a better gut microbiota to overcome some particular disease? So through the gut microbiota is one, and through is the second one is the through diet. How long I should take a better diet so that I will become healthy, or replenish the gut microbiota, or recover from IBD, IBS, or colon cancer? But what is your opinion? This actually question is because, especially when I give a talk on the probiotics, and we say that probiotics you should take 20 or 30 days, at least your biochemistry parameters will change. You will not suddenly become all right, but at least immunological response will change. Gut microbiota modulation will be there. So at least some changes we expect. With reference to the gut microbiota, means how many days you expect? One should be consuming either diet or have a good microflora in our gut. In other words, what is your opinion?
Okay, my opinion is straightforward. As you are the scientist, and you can find out the answer with that. I'm really love that finding that answers.
Yes.
In parallel to that, I want to make a comment based on my current work. So your question is that we have you have a questions like how exactly the gut microbiota treatment. Let's say, for example, if I have a drug treatment, you know very well that let's say, for example, if I have a normal a bacteremia or a normal infection, and you took a 4 days of antibiotic treatment, and it's over, and you will be feel better, right?
Exactly.
And yes, and your question is that if you are really treating let's say, for example, such kind of diseases with the help of microbiota, how long or how the efficiency or how efficiently we can treat with the period also, right? Right. Yes. So coming to the answer is that we are also working on the same line. So we have a clinical trial. Give me a second so I can share some screen.
Yeah. So when you are doing a clinical trial, what is your duration of study? In other words.
Okay. So okay. Okay. So this is like our clinical trial right now. Let's say, for example, we are targeting Oxalobacter formigens.
Okay.
And we wanted to make like whatever the oxalate level through the ingestion itself or through the diet itself, whether those kind of oxalates inside the gut can be decreased with the help of gut microbiota treatment. So so this is like a simple one. We have a 3 arms here. Let's say, for example, oxalate diet, low oxalate, high calcium diet. Then second one is the high oxalate, low calcium diet, and third one is that exactly the bacteria, Oxalobacter formigens. So, second, what happened? So in sorry. in our clinical trial, let's say, for example, we choose the population the healthy population, so who does not have the auxiliary vector formation colonization kind of things. So I mean to say, inside that gut, those are the healthy with the we check their healthy parameters like their blood parameters, their urine parameters, their all other healthy status. They are governing the healthy status there. So after that, we also have the inclusion criteria like it should not have the auxiliary vector colonization, auxiliary vector formation colonization inside the gut. So we choose those population, and we give them firstly as a low oxalate, high oxalate diet for the first week. Second week is the resting phase, and third week is like a high oxalate, low calcium diet.
Okay.
Then we observe the changes in the oxalate level into the urine as well as the fecal sample. So we believe that wherever there is a ingestion of oxalate diet or oxalic acid, it should reflect to the urine plus stool samples. Are you getting my point? Because those are the exogenous sources, right?
Yes.
So in our next phase, we wanted to give them as a treatment option like auxiliary vector formations who have ability to degrade oxalic acid. So with the help of that treatment, is it really reducing the oxalic acid levels into the urine itself? So whatever the reduction is happening by that bacterial intervention.
Okay.
Are you getting my point? So this is the whole story. But we find here that at an initial level, before the intervention start, we find the observation like there are the two populations observed. Like one population are the responder population, another is a responder non-responder population.
Yes.
What does that mean? That means that wherever there is a ingestion of oxalic acid, it should be changes of the oxalate into the urine or into the stool sample, right? So those are the responders. So we are expecting the responders, but half of the population is not responding. I mean to say, whatever you are giving the ingestion of the oxalic acid and the oxalate degrading specialist is missing
after some time.
But they are not responding to the oxalic acid. What does that mean? So whatever the non-responder bacteria are there, population are there, those are the oxalate level or the ingested oxalate is been completely removed by the bacteria from the gut itself, except oxalobacter formation. I mean to say that population does not have the oxalobacter formations. So we have a previous assumption that oxalobacter formations are the active metabolizer inside the gut, but we are into the additional knowledge like this is oxalate metabolism is not only related to the oxalobacter formations. There are number of species inside the gut. So I mean to say is that here, so we are able to formulate two logical phenomena like oxalate degrading plus oxalate degrading plus oxalate efflux activity by the effluxes into the feces promoting species into the gut itself. So this is a fantastic findings to us. So we can proceed with that, right?
Yeah, looks quite interesting. The very exhaustive study which you are representing here with using the different omic approaches, the analysis approaches, metabolite analysis, then the holomap study which you are actually taking up. I think that looks quite actually.
Yes.
There is a last question. Of course, that is a comment. A large number of still some publications are coming up with reference to the gut microbiota of tribal and then modern human being or the civilized person. So if literature perhaps if you can share will be wonderful. Because in US also some laboratories are there which they are actually using the tribal gut microbiota, which can be actually very suitable to treat several lifestyle diseases, and they are identifying a specific bacteria. Like I think in tribals from Indian tribals, they identified pivotal or dominance, So like this way, some studies, if it is there, if you can share, will be wonderful, will be educative for us. Since you are working in this area, I think you are really a resource person for us, for microbiology society in general, and several of our researchers, because I can see several questions were there from the other research around probiotics with relation to their health benefits of treating several diseases, and also it was a wonderful lecture. Now, thank you very much once again for answering all my comments. Yeah, thank you.
Thank you,
Thank
sir.
you very
Over
much,
to the
sir.
organizer. Yeah.
Can you please share your email ID in the chat box so that other participants, if in future wanted to contact you, they will contact. And really very nice interaction in today's guest lecture. Many more queries have been arised on the said topic. It has really emphasized and elaborative interaction, so that we people are really getting aware about the different factors which may affect the microbiota and its effect for the disease control and all those things. So really very nice interaction in today's guest lecture, sir. Thank you very much, sir. On behalf of the organizing committees, Honorable Principal Professor Mahendra Dhore, sir, Science College, Congress Nagar, Nagpur, and Honorable President, Microbiology Society, India, Dr. A.M. Deshmukh, sir. On behalf of the organizing committee, we are very much thankful that you have accepted our invitation for this precious guest lecture on the series Indians Speaking from Abroad. Thank you very much, sir.
Thank you, madam. Thank you for the nice opportunity. Thank you so much.
Yes,
I take a leave now.
yes, sir. Yes, thank you.